Hi everybody, this video will cover the following topics: DNA transcription and translation, exons and introns, promoters and operons, and protein creation in the ribosome. Your genes contain DNA, which have little bases or nucleotides on them that when read together create type of code to be read in order to make a protein. A protein is a molecule that carries out specific functions out in the cell, and are incredibly important for your body to maintain itself or do anything really. And each gene makes a protein. But the process doesn't just skip from DNA straight to protein. Rather, there's a step in the middle which includes the creation of something called mRNA. RNA stands for ribonucleic acid, and like DNA, has little bases on it. However, instead of thymine base, RNA uses uracil, or U. RNA is also single-stranded, so it looks like DNA, but it's cleaved in half. Ribosomes are organelles in the cell in the cytoplasm or on the rough endoplasmic reticulum, and they are what assembles and organizes proteins. Proteins are made of different amino acids and change the shape of the protein depending on what's used in the sequence. However, the problem is, DNA contains all the information for what amino acids make up each protein, and is stuck in the nucleus. So how do we get it over to the ribosome? The answer is RNA. RNA is made by splitting a specific sequence of DNA down the middle, typically the length of one gene. And then an enzyme called the RNA polymerase is connected to a strand. The polymerase will specifically attach to a sequence of bases called a promoter. This RNA polymerase will then copy the complementary bases onto a brand new piece of DNA called messenger RNA, or mRNA for short. However, not all of the mRNA contains what will leave the cell. Instead, the mRNA contains specific parts that we will cut out before it goes to code for a protein. The segments that we cut are called the introns, and the parts that we keep are called the exons. Exons contain the information that will actually be used in the ribosome. Once all the introns are cut out, the exons are able to assemble themselves in any order, meaning that there are a multitude of different combinations that they can take for the final mRNA strand they assemble. This makes it so that the same gene can code for multiple proteins in the end because of this exon variation. We call this alternative splicing. The mRNA is then equipped for a 5' G-cap, meaning that a guanine nucleotide is placed on the 5' end. A poly A tail is added to the 3' end, meaning that multiple adenine nucleotides are placed at the other end. Imagine these like protection gear. There are enzymes inside the cytoplasm that will degrade the mRNA, and these prevent the mRNA from getting damaged by them on its journey from the nucleus to the ribosome. That marks the end of transcription. Next, let's move on to translation, which is the process that happens for the mRNA to become a protein. Proteins are made of amino acids, and the ribosome essentially helps to assemble these amino acids to create them. The mRNA is made up of codons, and each codon is an amino acid. The specific order of bases that make up the codon determines what amino acid will be used to make up the protein. Next, let's move on to ribosomes. The mRNA strand will arrive at the ribosome. It will attach itself and move to the translation starting site. Then, molecules called transfer RNA, or tRNA, come to the ribosome to read the mRNA's codons. Each tRNA molecule carries a specific amino acid, and at the bottom has a complementary set of bases to a specific codon. So, if they correspond, the tRNA will then be able to attach itself. Then, it will drop off the amino acid and leave. The next tRNA molecule in line will then drop off its amino acid and leave, repeating until they eventually hit a stop codon in the mRNA. This sequence of amino acids will bind to each other, forming a protein. So essentially, we just took the information from the DNA, transcribed it onto RNA, and then translated that information into an actual protein. This forms what we call the central dogma, the idea where we go from DNA to RNA to protein. Lastly, let's move on to promoters and operons. Promoters are essentially the starting site for any transcription, and are a sequence of bases that signals polymerases to attach it to that site. Operons, on the other hand, control gene regulation. If a specific gene is always activated, it can lead to overproduction of a specific molecule and have serious consequences. On the other hand, if it is always deactivated, then the molecule that was supposed to be made from that gene will not be able to mate in times where it's needed. So, as a solution, we have things called operons on different genes. Note that operons are only for prokaryotes, but both eukaryotes and prokaryotes have promoters. Operons are essentially a sequence of bases that come after the promoter. There are two different types of operons, a repressible operon and an inducible operon. Repressible operons are when the gene is usually turned on, but we want to turn it off. A molecule called a repressor can bind to the operon, so that when RNA polymerase comes to make the protein the gene codes for, it is blocked by this molecule. Genes usually have the sequences to make these repressors a little before the promoter. The most commonly used example of a repressible operon is the TRP operon, which codes for enzymes that help to make the amino acid tryptophan. The TRP operon is by default inactive and does not prevent production. If there's an excess, it would be a waste to keep making more tryptophan, so we want to turn it off. Tryptophan is actually a co-repressor, meaning that it is the molecule that activates the repressor, which will then attach to the operon and prevent further transcription. Inducible operons are the opposite, where the genes are usually turned off, but we want to turn them on. The most commonly used example of inducible operon is the lac operon. The gene the lac operon is on codes for proteins that break down lactose, a type of sugar. However, sometimes we don't have a lot of lactose in our body, so we're making these proteins to break down nothing. So a repressor is needed in order to stop wasting resources. However, when there is actually lactose, what happens is the lactose will bind to the repressor, taking it off and allowing for the RNA polymerase to keep on going. Thank you for watching. If you have any feedback, please leave it in the comments below. And thank you everyone for watching.